I want to introduce you to our founder and CEO, John Santalucia.
Welcome to DNA Software. Let's go on a tour. We start our tour today at this site in front of this freezer, which is symbolic of a new chapter at DNA Software. So for 21 years, our company specialized in making diagnostics for other companies and doing custom research. And now we are at a new chapter where we are setting a goal to develop diagnostics for more than 300 human pathogens. One of the critical aspects of developing a high-quality diagnostic is to have a culture of cleanliness. So we separate the functions of each step of assay design in different rooms. This room is our validation preparation room. So we receive samples that we develop our PCR kits to test our PCR designs. We have a robot here that sets up the 96 well plates or 384 well plates, which we'll then do further PCR amplification on. After our plates are prepared in the robot room, we come to the PCR room where we have our instruments for running PCR. And the single purpose of this room is for these instruments to run the PCR reactions. Once our PCR reactions are completed, we go to this room to do post-PCR analysis. One of the questions we have is, did we form the correct amplicon? We use the tape station instrument here in order to analyze the amplicons to see if they are the correct length. In addition, we can check the amplicons to see if they are the correct sequence by doing nanopore sequencing, which we have in a different part of the facility. Next, we come to the next chapter in the history of DNA software, the development of our subsidiary company, FlexDX. FlexDX will do the kit manufacturing, marketing, and distribution of all the kits produced by our company. In this room, we receive oligonucleotides that we use to produce new kits for our products. So we can produce kits of around between 500 and 1,000 kits at a time. And we can use this robot over here to actually aliquot out those kits into individual batches. Or what's running right now is the robot is being used to pre-spot a 384 well plate with a series of assays that are for multiple patients. We have developed partnerships with a GMP and ISO certified facility called Hariba Incorporated, and they are going to be able to produce the kits on a much larger scale and distribute them from throughout the world.
So John, why don't you tell our customers more about what's happening in our new facility?
So what we are doing in this new facility, which we're going to move into August 1st, is organic chemistry. So in this room, we'll be able to synthesize new oligonucleotides with new modifieds in the backbone and in the bases, and also do oligonucleotide synthesis here to prototype new assay formats and new modified nucleotides. In this room, off of our organic chemistry facility, we will be expanding our instrument room, which will have additional PCR instruments for prototyping our assays. Thank you for joining us for this tour, and we look forward to serving you in the future.
Thanks for spending time with us and learning a little bit more about DNA software and FlexDX today.